Hi friends, in our previous video we studied how bilirubin is formed and how they are transported to the blood circulation. So when the bilirubin, that is unconjugated bilirubin, is formed, then this unconjugated bilirubin is sent into the circulation. For example, this is your blood circulation. When this unconjugated bilirubin reach into the blood circulation, they are highly lipophilic. Due to this lipophilic character, they can easily enter into the blood, into the body tissues. So some characteristic of the unconjugated bilirubin is that they are highly toxic, they are lipophilic, and they cannot pass into urine. Why? I will tell you. Okay. So after this, when the unconjugated bilirubin reach into the blood circulation from the liver, there is the albumin. This is formed by the liver, circulating into the blood circulation, highest concentration. This albumin is bind with the unconjugated bilirubin, and now this complex is called albumin bilirubin complex, and this complex is so big that it cannot cross the glomerular filtration. It cannot cross the kidney. Okay, it cannot pass into the kidney, or it cannot you can say pass into the kidney. Okay, cannot pass through the kidney, pass through kidney into urine. So urine is free of unconjugated bilirubin. So when this unconjugated bilirubin through the blood circulation reach to the liver, there present specific receptors for the liver for the uptake of unconjugated bilirubin. So for example, this is the liver. Here present specific type of receptors, transporters, specific transporters, and these transporters help in the transportation of unconjugated bilirubin into the liver. This is the unconjugated bilirubin. They transport these into the liver. Now it reach into the liver. The first step, that is the transportation of liver, or the uptake of liver by the hepatocytes or the liver. Okay, that is the first one. Uptake of liver, uptake by liver. Once the uptake of once the bilirubin, unconjugated bilirubin, is uptake by the liver with the help of specific transporter proteins, they enter into the hepatocytes. For example, this is our hepatocyte, and on the wall of the apical side of the hepatocyte present special transporters again, and these special transporters, they are called as ligands. These ligands proteins, they transport the unconjugated bilirubin into the hepatocyte. Now we reach into the hepatocyte cell. Once it reach into the hepatocyte cells, they are conjugated. They pass through the process of conjugation. This is in the rough endoplasmic reticulum. How the conjugation occurs, I will tell you. So the first step, that is the uptake by the liver through specific transporters. Once it reaches into the interstitium, they are taken up by the hepatocytes with the special receptor transporters. Ligandin transport the unconjugated from the parenchyma or from the interstitium into the hepatocytes. Once it reach into the hepatocytes, so number first uptake by the liver, number second uptake or entry into the hepatocytes. Entry into the hepatocyte. Okay, once it reach into the hepatocyte, for example here I will make a big hepatocyte. This is the nucleus of hepatocyte, and here present rough endoplasmic reticulum. In the rough endoplasmic reticulum, the unconjugated bilirubin, they enter into the hepatocyte and into the rough endoplasmic reticulum. Here present specific enzymes, and these enzymes, they are responsible for the conversion of unconjugated bilirubin into the conjugate bilirubin. So these enzymes, what you call is UDP, glucuronide transferase. So this UDP glucuronide transferase is enzyme which help in the conjugation of bilirubin. So now let us discuss the process in detail. That, for example, unconjugated bilirubin reach into the rough endoplasmic reticulum. There present specific enzymes, and these specific enzymes called is UDP glucuronide transferase. What they do, they aid glucuronic acid to the unconjugated bilirubin. Now it become conjugated bilirubin. So how this process occur? For example, this is unconjugated bilirubin, and this enzyme, that is UDP glucuronide transferase, they transfer glucuronic acid to the unconjugated form. In the initial, it added only one glucuronic acid. Now then it can become the bilirubin monoglucuronate, that is bilirubin monoglucuronate. After the addition of another glucuronic acid by the enzyme glucuronide transferase, it converts into bilirubin diglucuronate, and then these are excreted into the bile. So for example, now let's make hepatocyte again. Okay, this is the hepatocyte. Bilirubin unconjugated enter then this unconjugated bilirubin convert into two forms. Number 1, bilirubin monoglucuronate. Number 2, bilirubin diglucuronate with the help of glucuronyl transferase enzyme, and very little amount still remain as a unconjugated form. So this is conjugated bilirubin and this is also conjugated bilirubin. Now after the conjugation process, okay, this is what conjugation process. Conjugation. After the conjugation, these bilirubin, conjugated bilirubin, they are excreted into the biliary canaliculi. So here the specific enzymes transporters are present and they are responsible for the excretion of conjugated bilirubin. And these enzymes what we called as multi-drug resistant associated protein type 2. This multi-drug resistant associated protein type 2, they are responsible for the removal of BMG, PDG and very little amount of unconjugated bilirubin into the biliary canaliculi. Estimately, we can say that 90% excretion occurs of bilirubin diglucuronate, 7% bilirubin monoglucuronate, and very less than 5% of unconjugated bilirubin. So this is about the excretion and conjugation. A disease related to that of the multi-drug resistant protein we will discuss later, that the diseases in which the mutation occur in the BMD RP protein and also mutation occur into glucuronyl transferase which leads to intrahepatic jaundice and obstructive jaundice. So we will discuss these type of disorders later on. Thank you.